Good morning, students. Welcome back to biology class, chapter 9, Biomolecules, Nucleic Acids. Today's class, we are going to discuss about the nucleic acids. In 1869, nucleic acids was discovered by Frederick Michaud, and he gave the name nuclein because it was located in the nucleus. Later, Altman gave the name nucleic acid because of the presence of phosphoric acid. Nucleic acids are present in the cytoplasm of the cell organelles like mitochondria, chloroplast, etc. There are two types of nucleic acids are present. The first one, deoxyribonucleic acid, DNA. Second one, ribonucleic acid, RNA. Now we'll discuss about the chemical composition of the nucleic acids. Nucleic acids, DNA and RNA, consist of a sugar molecule. If it is a DNA, deoxyribosugar. If it is RNA, ribosugar. It is a 5-carbon compound, pentose sugar. DNA contains a deoxyribosugar and the RNA contains the ribosugar. Then the second chemical composition is the nitrogen bases. Nitrogen bases are divided into two groups. The first one, it is the purine groups that contains adenine and guanine. The second one, it is the pyrimidine, thymine and cytosine contains. Okay, nitrogen bases are divided into two groups, purine bases and pyrimidine bases. Purine bases contains the adenine and guanine. Pyrimidine bases contains the in DNA, thymine and cytosine, in RNA, uracil and cytosine. Thymine is present only in the DNA, uracil is present only in the RNA. So the nitrogen bases in the DNA are adenine, guanine, thymine and cytosine. In RNA it is adenine, guanine, cytosine and uracil. Then one phosphate group, that is the phosphoric acid, is present. So these are the chemical composition: sugar, nitrogen bases and the phosphate group. Now we will discuss about the nucleoside. Nucleoside means nitrogen base plus sugar is a nucleoside. First we'll go through the adenine base. Adenine plus deoxyribosugar, deoxyadenosine. Guanine plus deoxyribose, deoxyguanosine. Thymine plus deoxyribose, deoxythymidine. Thymine plus deoxyribosugar, deoxythymidine. Cytosine plus deoxyribosugar, deoxycytidine. In RNA, adenine plus ribose, deoxyadenosine. Guanine plus ribose, deoxyguanosine. Uracil plus ribose, uridine. And last one, it is cytosine plus ribose, cytidine or cytosine. Now we'll discuss about the nucleotide. Nucleotides are the formation of the nucleoside plus phosphoric acid. A nucleotide is forming. When a phosphoric acid is linked with the 5-carbon pentose sugar of the nucleoside, the nucleotide is forming. Okay, then it is the DNA, it is the deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate and deoxycytidine or cytosine monophosphate. Then in RNA, adenosine monophosphate, guanosine monophosphate, cytosine monophosphate and uridine monophosphate. A nucleotide with one phosphoric acid is called nucleoside monophosphate, and the two phosphoric acid is called nucleoside diphosphate. 3 phosphoric acid is called nucleoside triphosphate. Okay, these are the, or we can call it as adenylic acid, guanylic acid, thymidyllic acid and cytidyllic acid. These are the nucleotides present in the DNA. Adenylic acid, guanylic acid, cytidyllic acid and uridylic acid. These are the nucleotides present in the RNA. Now we will discuss about the structure of the DNA. It is a double-stranded structure. Double-stranded means that contains the polynucleotides. Double-stranded structure, it contains the polynucleotides from that is arranged with spirally arranged. One is the phosphoric acid in the middle and the sugar and phosphate are present in the backbone of the polynucleotide chain. Nitrogenous bases is arranged inside the structure. Okay, the base and the sugar base is present in the middle and sugar and phosphate in the backbone. It is a double stranded structure, and the starting with 5 prime to 3 prime. One strand is 5 prime to 3 prime and the other strand is 3 prime to 5 prime connected by the glycosidic bonds. So it is 5 prime to 3 prime, 3 prime to 5 prime. So it is called anti-parallel. See that one, the DNA structure is double-stranded, helically coiled structure, and it is contains the anti-parallel. Then the base pairs are, see that one, the base pairing. The one of the strand is 5 prime to 3 prime strand is made up with the purine, and it may, the other strand 3 prime to 5 prime is always, it is a pyrimidine strand, or vice versa. Means 3 prime to 5 prime is purine basis, or the pyrimidine basis, or that time it is purine basis. Okay, and it is always adenine is paired with thymine by 2 hydrogen bonds, and the guanine is paired with cytosine by 3 hydrogen bonds. See that one, a purine paired with the pyrimidine by hydrogen bonds. Example, adenine is paired with the thymine by 2 hydrogen bonds, and the guanine is paired with the cytosine by 3 hydrogen bonds. This is called complementary base pairing. Base pairing, one purine bases are paired with the pyrimidine bases by hydrogen bonds is called base pairing, complementary base pairing. Now, next we will see the Chargaff's base equivalence rule. Chargaff's base equivalence rule means that is the amount of purine bases are equal to the amount of the pyrimidine bases. Adenine plus guanine is equal to the cytosine plus guanine. See that one, adenine plus guanine is equal to guanine plus cytosine. That is equal. So one of the strand is if you know that one of the element we know that one, then we can find out the other one. One example, a DNA sample contains 30% of adenine. What will be the percentage of guanine in the given sample? According to the Chargaff's rule, you see that one, adenine plus thymine is, adenine plus guanine is equal to thymine plus cytosine. Adenine is equal to thymine, guanine is equal to cytosine. According to that, the answer is 20%. We will get that answer. Now we should see what are the differences between DNA and RNA. In the first DNA, the sugar present in the deoxyribose. RNA, the sugar present is ribose. Nitrogenous bases are adenine, guanine, thymine and cytosine. In the RNA, nitrogenous bases are adenine, guanine, cytosine and uracil. and the DNA is double-stranded. RNA is a single-stranded. RNA is always single-stranded. DNA is the genetic material of almost all the living organisms. RNA is the genetic material of only some of the viruses. And it is DNA is chemically less reactive and structurally more stable. But in the RNA is chemically more reactive and structurally less stable. DNA is only one group, one type. RNAs, there are two types. Mainly it is genetic and non-genetic RNAs are there. Genetic, it is in the viruses it is present. Non-genetic RNAs are divided into three types. mRNA, that means messenger RNA, tRNA means transfer RNA, and rRNA, ribosomal RNA. Now we'll see the chemical nature and role of ATP and NAD. Adenosine triphosphate. ATP is a mononucleotide, has three parts connected by covalent bonds: adenine, a nitrogen base, ribose, a 5-carbon sugar, and 3 phosphates. Two covalent bonds linking the 3 phosphate together are called high energy bonds. Thank you.